Hey, ladies and gentlemen, welcome to Chapter 5 Biology. We are going into DNA, RNA, and protein synthesis. All right, that is three things in this chapter. DNA, you should probably already know about. That is your genetic code. It says how to build your body. RNA, you may have heard of. It's what actually does the building. So DNA is like, this is how you build a body. RNA is like, got it, guys, get to work. And then they start trying to build your body. Now think back to chapter 1. I told you what your body is made out of: proteins. So DNA says how to make proteins. RNA is going to make proteins, and that's what this chapter is going to be about. Now, this is going to be a very large chapter, fair warning. In a normal school year, I would cover DNA as its own kind of standalone thing before Christmas. We would go on Christmas break, come back, and then do RNA as its own other chapter. I don't know how things are going to line up this year, but it's about 2 chapters worth of information. So let's get started. We have about 11 vocab words to talk about. So like I said, we are talking about DNA. What these letters stand for, DNA, is deoxyribonucleic acid. Now, what does those words mean? Deoxyribonucleic acid. All right, well, you should already know nucleic acid. Think back to chapter 2. We said we have 4 macromolecules: right? Carbohydrates, proteins, lipids, and nucleic acids. That's the Na, right? Deoxyribonucleic acid is just a kind of nucleic acid. So what is DNA? It is a macromolecule with genetic information and is capable of self-replication and the creation of RNA. Oh, that is a mouthful. I don't like that definition, guys. That's your keystone definition. Let me simplify this, all right? This picture. This one's easier to look at, but this one's more accurate, right? This picture is DNA. And if you look at it, it just looks like a whole bunch of circles, right? Let's zoom in on these circles, though. Take a look at this. You are looking at molecules, monomers. Remember chapter 2, get a bunch of monomers together to make your macromolecule. And remember, molecules are made out of atoms. So these little circles are all different atoms. See these 3 dotted lines here? That's a separation. So this is one molecule. This is another molecule. This is a molecule. And this is a molecule. These molecules are called nucleotides, and there are only 4 in DNA. We call them adenine, thymine, cytosine, and guanine. Since your DNA is genetic information, guys, it's a message. Think of this as words, and these are the letters. These are the letters of your alphabet. Your DNA only has 4 letters of the alphabet: adenine, thymine, cytosine, and guanine. Let's just call them A, T, C, and G for short. A, T, C, and G are your only 4 letters of the alphabet. And when you put them together in different ways, you make codes. And those codes tell you how to make your body. DNA is basically a biological flash drive full of information. That's how you're going to think of it. This chapter is going to be about how to read the information on this flash drive. Nucleotide, that's actually what we just looked at. A monomer. These things. Made of sugar, phosphate, and the base used to make nucleic acids. So this looks much simpler, right? A circle, a pentagon, and a rectangle, right? That's what we were looking at over here. This one is more detailed. This is the circle. It's a phosphate. Here is that pentagon. It's a sugar. And this part right here is the base. Okay. The phosphates and the sugars are exactly the same in all 4 of them, so we don't need to learn anything new about them. But the bases in the middle, this rectangle part, that's what's different over here. So take a look. We have a hexagon shape and another hexagon shape, and then a hexagon with a pentagon and a hexagon with a pentagon. So there are already 2 different categories. This hexagon shape has a little gray circle with white circles attached. This hexagon shape does not. It has a blue one on the other side. This hexagon with a pentagon has a blue one with white ones, and this hexagon and pentagon does not. It has a little red one. So these are 4 different molecules creating different messages in the DNA. We call those bases. The phosphate, the sugar, and the base make up one nucleotide. Deoxyribose, the sugar molecule in DNA. That was simple. This thing right here, the sugar in the nucleotide. It's called deoxyribose, which is where this name comes from. Deoxyribonucleic acid. It's a nucleic acid with a deoxyribose sugar. So if you look at the DNA unraveled, turn your head to the right, and you'll see a phosphate, a sugar, and a base. Just like right here, phosphate, sugar, base. The deoxyribose sugars are just a backbone, a structure to hold the bases in place. So it's the bases that are important. The deoxyribose sugar just keeps them there. Double helix, the twisted ladder shape of DNA. Once you get all these bases together, notice that these don't really look like they would fit together perfectly like puzzle pieces, right? They're kind of a hot mess. Well, they don't fit together perfectly. When you actually get them together, they have to twist into place, and all of them twisting into place causes the entire DNA strand to twist up, and you get this shape that we call a double helix. Helix means it's twisting, and double means that there are two sides to the twist. Double helix, twisted ladder shape of DNA. The sugar and the phosphates twist around the base pairs. DNA replication, the process in which DNA makes a copy of itself. So this is how you make new DNA. So taking a look at this GIF, we have the double helix right here, and it's getting split into two halves. It's colored in blue and red. The blue goes one side, red goes the other side. And because of those base pairs, your body knows how to fill in the gaps. Turns out they match up in a way. You have the A's, T's, C's, and G's. So if there's a blue A right here, then a red T is going to match up because A's and T's go together. And if you have a blue C, then a red G is going to go there because C's and G's go together. And we end up getting the reds and blues back together exactly the way it looked over here. And same thing down here, we are going to get reds and blues back together here exactly the way it looked over here. We end up with two double helices that is completely identical to the original double helix. That is called DNA replication. Number 6 is kind of confusing. The semi-conservative model. This is the correct way DNA replicates. Why am I specifying that this is the correct way? Because there's a little bit of history that may or may not come up on the keystones. So let me do a little history lesson for you. Once upon a time, people didn't know how DNA replicated. They knew that it did, but they had no idea that it looked like this. A bunch of different scientists were arguing with each other and came up with 3 different possible models. Semi-conservative, which we just saw, the DNA splits in half and new nucleotides move in to complete the base pairs. Another group of scientists came up with what they called the conservative model. They thought your DNA didn't split apart and somehow your body just knew to make a photocopy based on how it looked with no explanation of how that figured that one out. And then the dispersive model, which instead of splitting down the middle, they thought it split in chunks and then new pieces just filled in the gaps somehow without creating mutations with no explanation of that one. So needless to say, conservative and dispersive were both proven completely wrong. Semi-conservative actually has been proven correct. This is no longer a hypothesis or even a theory. This is a fact. Chargaff's rule. Chargaff was a scientist studying DNA, and he came up with a very simple rule based on another existing rule. The existing rule is the one I told you, that A's and T's, look at this picture in the middle here, A's and T's always pair together. C's and G's always pair together. Same thing here, C and a G and an A and a T, always pair up. So his rule based on this is that in DNA, the number of adenines, the A's, adenine bases is equal to the number of thymine bases. And the number of cytosine bases is equal to the number of guanine bases. What does that mean? It means if A's and T's always go together, if you have 100 A's, then you have 100 T's. Right? Think of it like shoes. You have a left shoe and a right shoe. If for some reason you had 250 left shoes, then you also have 250 right shoes because they come in pairs. A's and T's come in pairs. So the numbers are the same. Why is that important? Because we're going to be doing math based on this. Not today, though. That was the first half, all about DNA. Now we're going to be talking about RNA and how RNA is used to make proteins. So what is RNA? It's what we call ribonucleic acid. DNA was deoxyribo. This is ribonucleic acid. A single-stranded nucleic acid that reads DNA to make proteins. Here is a picture of DNA, double helix. RNA is a single helix. In fact, it doesn't even have to be a helix. It can just be bent around itself. How is it made? That's very important, guys. Think back to chapter 1. Why do I keep going back to chapter 1 and chapter 2? Because you're going to be seeing these two chapters for the rest of the school year. In chapter 1, we talked about this is the cell, and here in the middle with these holes in it, that is the nucleus. And inside the nucleus is the nucleolus. The nucleolus makes pieces of RNA. It makes RNA nucleotides, phosphate sugars and bases, right? This is an RNA nucleotide. And then those RNA nucleotides are going to leave the nucleolus and float around the nucleus. Who can remember what else is inside the nucleus of the cell? If you guessed DNA, congratulations, you are correct. And that DNA is going to get opened up. That's the blue, by the way. Blue is DNA. And then the red represents the RNA nucleotides pairing up individually. Look at C with a G. A pairs with a T. And this one's weird. RNA doesn't have T's, so they have a U instead. So A with U pairs with an A. Just treat U's like T's. And they end up pairing up to make a copy of the code. Look up here, C-A-T-T-C, C-A-U-U-C. Remember, treat U's like T's, C-A-T, C-A-U, T-C-G, U-C-G. It's the exact same code. Then this code, now in yellow for some reason, is going to leave the DNA and is going to, there it is down here at the bottom, is going to be used to make a protein. This right here, that's what's being made, a protein. So what are these yellow things coming in? That's another kind of RNA. That kind of RNA has the protein pieces. The RNA we just made says what order to go in. The yellow ones read the code to bring the pieces in and assemble the pieces in the right order to make a protein. Codon and anticodon. Codon, 3 nucleotide sequence of mRNA that codes for a single amino acid. That red line that we made is now down here in black, right? That's called mRNA. And every 3 bases is called a codon because it is a code. It is asking for a specific amino acid. That's a piece of a protein, by the way. Chapter 2, guys, proteins are made out of amino acids. These circles are amino acids. And in every, zooming in, every 3 codes, UAC, that's one codon. GAU is another codon. So a codon is asking for the amino acid. Anticodon, that is what's on what we call the tRNA, a group of 3 nucleotides on the tRNA that carries the amino acid. Codon is asking for the amino acid. Anticodon has the amino acid. So the anticodon is going to be GGC, so it's looking for a CCG. And it's going to put that amino acid in that spot. And then the ribosome in the background puts them together. I understand that's a bit confusing, guys, especially with me going back and forth between the pictures. So let me do it one more time with just this simple picture. This is, follow my mouse here. This black line is the mRNA that we made. one, two, three nucleotides is a codon. And then 1, 2, 3 nucleotides on the tRNA is the anticodon. The anticodon pairs with the codon. A's with U's, C's with G's, remember that? And this tRNA is going to bring this yellow dot here, zoom it in, that yellow dot right there, is an amino acid. It is going to put the amino acid in the correct spot because the anticodon paired with the codon. When all the dots are in the correct order, we've made a protein. That was really complicated, guys. That is going to be multiple days of learning. If you don't get it now, that's fine. Just have these definitions memorized. And our last word today is chromosomes, condensed DNA in the nucleus of a cell. Humans have 46 of them per cell. Right. Common misconception, guys. Every year I have a lot of students who think you have one strand of DNA, like one strand floating around your body. That's it. If you lost your DNA, that's it. You're done for. No, that is very wrong. Every single cell in your body has a copy of the DNA, with the exception of your red blood cells. Worry about that in anatomy and physiology. Every other cell in your body has a copy of the DNA. If you're going to try to fit an entire strand of DNA, which is the longest molecule in your body, into one cell, that is going to be a mess. Like, try to stick your headphones into a drawer. What's going to happen? Those headphones are going to come back tangled, right? Same thing with your DNA. If you're going to stick all of that into one cell, that's going to become a tangled mess. So your body is going to do this neatly. You are actually going to take this one strand of DNA and chop it. That's right. You are going to cut your DNA into 46 pieces. And then those pieces are going to get very neatly braided up. And that's what we call chromosomes. 46 neatly braided chunks of DNA. And every cell in your body has them. Now, why do we do that? Why do we care about keeping them nice and neat? Because in a future chapter, we're going to talk about mitosis, which is where your cells are going to split down the middle and become two cells. So take a look inside this nucleus. You see a hot mess of DNA floating around, but uh-oh, see that? They just organized. Now those organized pieces can very neatly get separated. So that, and then they become an untangled mess again. So again, tangled mess, and then boom, nice and neat. Those neat pieces get separated and taken care of. So that is why we do this. All right, that is it for the vocabulary. We are going to go into much more detail about all of this stuff when we get into the notes. So until then, go back in Google Classroom, see if I left you any work to do, and I will see you in the next video.